Good afternoon, everybody. This is Nitya Shri S from Saptagiri College of Engineering, Department of Biotechnology, Bangalore. My topic of discussion is metabolomics and air in cyanide toxicity. Next slide, please. First, let us see what is metabolomics. Metabolomics is the study of small molecules or metabolites in the biological sample. So the actual definition is the systematic study of the unique chemical fingerprints that specific cellular processes leave behind. So can you please click? The factors which affect the metabolomics are genetic and epigenetic factors, lifestyle, nutrition, drugs, diseases, environment, age, etc. And these factors will affect the DNA methylation and histone post-translation factors, which indeed modifies the phenotype and genes. So why metabolomics is necessary? It is necessary for analyzing the metabolic pathways which are targeted by the harmful chemicals, and it helps in the prediction of drug responses. It gives clarity on the pathological mechanisms in discovering novel drug targets, and metabolites acts as biomarkers for diseases. So metabolomics is necessary. And overview of the metabolomics is the DNA is transcribed into RNA, and the RNA is translated into protein, and this as a whole is called as transcriptomics. And the quaternary structure of protein interacts with another protein, and this interaction leads to a release of metabolites, and the study of metabolites is called as metabolomics. And keeping metabolomic studies as the base, many toxic chemicals, metabolites, and its interruptions in metabolic pathways are studied and analyzed. This is used in the identification of cause of death. So one such toxic chemicals which we are going to discuss is cyanide poisoning, along with a case study. So cyanide is known as a fatal poison, and cyanide is the cyanide is a chemical which has carbon and nitrogen bonds, and it is a negatively charged ion. So cyanide has its derivatives like in the vapor phase, it is hydrogen cyanide, which is known to be very fatal. And in the solid state, it is potassium cyanide and sodium cyanide. The mechanism of cyanide toxicity is that the cyanide binds to the ion atom in cytochrome C oxidase in the mitochondria of cells during ETC cycle. So this cyanide is a irreversible enzyme inhibitor which prevents the transportation of electrons to oxygen, and the ATP is not synthesized, and the muscles which is in the need of ATP will not get the oxygen which is synthesized, and hence the cell undergoes apoptosis. So when there is large amount of cells undergoing apoptosis, the victim dies. So the structure of cyanide is that the carbon is bonded to nitrogen with 3 hydrogen bonds. And signs and symptoms: dizziness, headache, nausea and vomiting, diaphoresis, which means excessive sweating, rapid breathing and heart rate, loss of consciousness, low blood pressure. And the toxic dose of hydrogen cyanide is 60 to 90 milligrams, while that of potassium cyanide and sodium cyanide is 300 milligrams. So next slide, please. So coming to the case study, we consider 41-year-old man who was found to be unconscious in the metal chrome plating shop for a duration of time, and emergency medical services arrived and found the patient to be asystolic and started advanced cardiac life support protocol, and intubation was provided, and the patient was hypotensive, requiring dopamine and norepinephrine. Dopamine is a neurotransmitter, and norepinephrine is responsible for blood pressures. And due to the work environment of the patient, he was provided with cyanide antidote kit, which consisted of sodium thiosulfate 12.5 grams and sodium nitrite 300 milligrams, 14 minutes after the arrival at the emergency. The reactions in the patient were the brain death protocol had already begun and CNS was not administered because the body temperature and blood pressure was almost normal. And later, when neurological examinations were showed the limited reflexes and apnea test was done, which failed. And due to the failure of apnea test, the patient was declared to be dead. And the samples collected were blood and urine. And the detection techniques were HPLC and GCMS. The postmortem appearances are cyanosed face and lips, where we can observe the blue, the face and lips have turned into blue color and red bruises all over the body and dilated pupils. Medical forensic examinations are the stomach, brain, liver, and so on have turned into bright red color due to lactic acidosis and ciliary loss of tonsils, cyanosis of face and lips, foam out of the body, and bruised corpse. Next slide, please. Metabolomics methods and analysis. This methodology is based on the sample which is collected. So urine, when urine is collected, 100 millimolar of sodium hydroxide is added and filtered using cartridge 2H and syringe filter, and it is diluted with deionized water in the ratio 1:3, and the cyanide is determined using ICPAD, which is ion chromatography pulsed amphoteric detection. And when the sample is saliva, then the empty vial is weighed first, and the vial and the sample is collected. Vial is weighed with the sample, and 100 millimolar of NaOH is added. Sample is diluted with deionized water to 2 ml. Filtration is carried out by cartridge 2H and syringe filter, and cyanide is determined by ICPAD. When the sample is sweat, the same procedure of saliva is carried out. And this is a graphical representation with different samples. Retention time versus height is plotted, and this graph shows the presence of cyanide in the patient. So a click. By plotting the graph of the area below the peak and the standard concentrations, it gives a linear curve, and this represents the correlation coefficient. Correlation coefficient values must be from -1 to +1, and as the correlation coefficient value increases, it determines the strength of the concentration of metabolites in the patient. Discussion and conclusion. The treatment basically depends on severity of symptoms, history of the patient, route of exposure, and what is available. And the chemicals used in treatment are sodium nitrite, sodium thiosulfate, sodium thiopentone, amyl nitrite, activated charcoal, supplemented oxygen, and hydroxycobalamin. And the cyanide cannot be left in the left as free in the environment in neutral pH because it gets volatilized into hydrogen cyanide, and oxidizing agents decompose the cyanide, and hence it is stored in polypropylene tubes, which is protected from light and is kept in the temperature -15 degrees Celsius until the analysis is made. These conditions are applied, and the conclusion is that the treatment was given to the patient intravenously, and the treatment was not given in the right time. That is, the cyanide antidote kit was not given in the right time, and hence the metabolite concentration increased in the patient, which led to the death of the patient. So, appropriate treatment using sodium thiopentone or thiosulfate is an emerging procedure and helps in the preventing of deaths due to cyanide poisoning. Thank you so much.